Hello guys, welcome to the knowledge domain, the concepts of life sciences and biotechnology. Today we are going to learn non-coding regulatory elements or the on, off and the dimer switches of a gene. Regulatory sequence can be located on either end of the gene or within. There are the three major types of the regulatory sequences or the regulatory portion of the DNA of a gene. So one is the promoter that we have already discussed in one of the previous lecture. There is the enhancer and the silencer. So in this lecture today we are going to focus on enhancer and the silencers. So regulatory elements which are necessary in controlling the expression of the gene, where, when, and how for the actively transcription of the gene. When these regulatory elements are present on the same chromosome as the gene, these are called the cis regulatory elements. So the promoter function is, we have discussed in the one of the previous lecture also, it is a place where the RNA polymerase it binds to initiate the transcription or the expression of a gene or to prepare the messenger RNA. Promoters are typically immediately upstream site of the RNA polymerase II binding site. This is around 1000 base pair and reach in the CPG. So the amount of the CPG is more than 50% in this area. So this is also called the CPG islands. Enhancer control the efficiency and the rate of transcription from a specific promoter. In contrast, the silencer or the repressor, it inhibits the expression or the transcription process from the promoter. Then what are the transcription factors? These are the proteins that binds DNA with precise sequence recognition for specificity. So these are the proteins that bind at the specific areas of the DNA, like they have to bind at the promoter sites. That they can bind at the enhancer sites or they can bind at the silencer site. So exact at the specific location, which is identified based on the sequence of the sequence of the DNA at that area. The transcription factor, it recruits nucleosome modified proteins to that region of the genome where it binds and make the chromatin more accessible for the RNA polymerase II to carry out the transcription. Transcription factor can recruit transcriptional co-regulators to form bridges which loop the chromatin that we are going to discuss in one of the following slides in a conformation that brings the enhancer bound transcription factor into the vicinity of the promoter. So these are the core regulator that is bringing the promoter and enhancer close to each other so that there is a tight regulation of the gene. The transcriptional co-regulators can be activated. They can be the repressor of the transcription. Then enhancers generally activate only the cis-linked promoters. Moreover, enhancer do not need to be on the 5' upstream. Means it can be found at any site, at the 5', at the 3', and at the introns also. So it is not necessary that enhancer are usually found at the 5' site. They can be found anywhere of the gene. It may be at the 3' end, it may be within the gene, in the intron region also. Enhancer DNA sequence are the same in every cell type, and enhancers and silencers both are modular in nature. In each cell, the enhancer becomes associated with particular transcription factors that bound nucleosome regulators and mediator complex, engages with the promoter to transcribe the gene in that particular type of the cell. This is an example. Here we can see this is the gene A. This is the gene A. This is the exons and the introns. These are the introns, these are the exons. So this is the part of the gene. It has two enhancers. One is specific to the limb, another is specific to the brain. So this looping of the chromatin so that this brain specific enhancer it is coming close to the promoter of the gene A in the brain specific, whereas the limb specific it is coming close to the promoter in the only the limb that we are going to discuss. So the specific combination of the transcription factors allow gene to be active in specific cell types. This is one of the example of the mouse P6 gene, and there are the several enhancers for the same gene. And this is also the principle of the enhancer modularity. So in this case, we can say that for the one gene, that is the PEX gene, we have the expression in the pancreas, the lens and the corona of the eye. This is the enhancer. This is the enhancer type in the neural tube, and this is the enhancer in the retina. And these eye are the ectodermal in origin. So this PEX gene has pancreas specific enhancer, lens and the corona specific enhancer, neural tube specific enhancer, and the retina specific enhancer. And how these enhancers are regulating into specific tissues because of the looping of this chromosome that is only possible because of the transcription factor that is bringing the enhancers close to the promoters. So the genes having multiple separate enhancer allow a protein to be expressed in several different tissues, but not expressed at all in others. So this is one of the example of how the chromosome is looping to bring the promoter close to the enhancer. So the function of the transcription factors is to create a bridge between the enhancer and the promoter. So this is the promoter and this is the enhancer. So this enhancer also called the cis regulatory element because usually they are present on the same chromosome where the genes are located. So there are two types of the transcription factors in this case. In fact, there are the three. So in this photo, there are the two. The one is the basal transcription factor that is binding at the promoter site so that RNA polymerase is going to means this the binding of the RNA polymerase 2 at the promoter site is facilitated or it is made possible because of the basal transcription factor. So that is also called the saddle. And when the RNA polymerase 2 binds, when the basal transcription factor is already present there, it is able to initiate the transcription. Then there are the other transcription factors, they are going to bind at the enhancer sites. And they are going to regulate when and where the transcription can occur. So this is how the loop is formed. So other transcription regulators do not bind to the DNA. They are going to bind with the enhancer that is bound at the enhancer site, the transcription factor, and the transcription factor that is going to bound at the promoter site. So these are the other transcription factors. They are going to make a bridge between the enhancer and the promoter, or it is making a link. So the chromatin it loops mean they are in the beginning it is a straight line, now it is a loop so that the enhancer and promoter can be brought together, and this is only possible because of the transcription factor. So the transcription factors it is assembling at the enhancer site but not till now the promoter is not in use. Promoter can only be used when the GATA1 transcription factor binds to the promoter. So this is the GATA1. When it is going to the bound to the promoter and the enhancer, then the promoter is in use. So the GATA1 recruit the LDB1. So you can see this is the LDB1. So it forms the bridge between the transcription factor associated with enhancer and the transcription factor associated with the promoter. So On the enhancers, there are the questions like what are the consequences of enhancer modularity to a developing individual or to a species? How might a mutation in an enhancer affect development? And because of this, what might occur in an embryo? If there were a mutation in the enhancer region of the gene, and could such a mutation evolutionary important? Yes, it does. So when there is a mutation in the enhancer region, it does affect the evolutions of that species or the development of the species. So ultimately, it has an the evolutionary effect. Next is the silencers or repressor or the negative enhancer, it is also known as. They actively repress the transcription, silence gene expression specifically in different cell types and temporally, means according to the time. In the mouse DNA sequence, it prevents promoter activation in any tissue, for example, neuron. And this is called the neural restrictive silencer element, NRSC. Several mouse gene expression is limited to the nervous system, like synapsin, sodium channel type 2, brain-derived neurotrophic factor, NGCAM, and the L1. The protein that binds to NRSC are called the neural restrictive silencer factor, or NRSF. NRSF appears to be expressed in every cell that is not a mature neuron. NRSC is deleted from the particular neuron gene, then there is a gene expressed in the non-neuronal cells. So this is a transgene. So in this one, it is a experimentally designed situation where we have the L1 promoter. It is with the log Z, and this is the NRSC sequence. So in this case, this is the NRSC sequence. It is replacing the expression of log Z. So you don't see anything pigmentation here. But when this repressor NRSC sequence is missing in this case, so the expression of the gene is not repressed. So we can see the beta galactosidase, the log Z product is expressed in the non-neuronal cell. In this case, it is only the neuronal cells because in this NRSC sequence is replacing in the other cell types and it is not present in the other cells, so it is expressing in the non-neuronal cell. This is the mouse embryo. Thank you.